So we'll go ahead and get started. Welcome. My name is Ian Thomas Binyami. I'm the VP of Member Ed for MDBA, and I'm really excited to welcome you to this February Zoom speaker event. Before we get started and before I introduce our guest, I wanted to just say a few words about some upcoming opportunities and events within MDBA. So let me cycle through a few slides here, and then I will introduce Dr. Alison McAfee, and she's going to lead us through her presentation. So one thing I wanted to mention, since we have an audience of engaged MDBA members here, is we're always looking for volunteers, and I know Sheila Weston, our VP of Community Ed, is always looking for people who are willing to help out, taking observation hives to elementary schools, to science fairs, to different outreach programs. So if you're interested at all in doing that, please go to the website, sign in. You'll be able to see the event calendar there. And on the event calendar, Sheila has posted the different MDBA volunteering events that are coming up. And we do a lot. I think there's like 5 or 6 just in the next couple of weeks. So she needs volunteers to do that. You can go to the event calendar and you can sign up there, or you can contact her directly if you have questions about volunteering. So I'd encourage you to do that. I also wanted to make a quick announcement about some upcoming meetings and events. The first is that the annual spring barbecue is happening on March 12th. That's our next general meeting. We'll have the barbecue and the raffle. An RSVP is required, so please log into the website, register for the meeting. And if you're not able to do that on the website, you can also contact our secretary, Andrea, and she can put you on the list, and you can pay at the door. So that event is coming up right around the corner. And finally, I wanted to let you know about 2 upcoming Zoom speaker events, our next two after tonight's presentation by Dr. McAfee. The first is Julia Mahood, who is the president of the Georgia Beekeepers Association, and knows a lot about drones and is really interested in drones and is the creator of the Map My Drone, mydca.com, which is a fun sort of citizen science project online about drone congregation areas. She'll be joining us on March 26th at 7 p.m. And then we also in April, we have Dr. Marla Spivak, who is an expert on hygienic behavior, who's going to be talking about hygienic behavior this April 23rd at 6 p.m. So hope you're able to join us for those events as we head into the rest of the year. It's my pleasure to introduce our speaker tonight, Dr. Alison McAfee. I'll say a few words about her background, and then I'll turn it over to you. Dr. McAfee has been researching honeybees for 12 years. She obtained her PhD in genomic science and technology from the University of British Columbia, where she graduated at the top of her faculty and went on to become one of 15 global winners of the L'Oreal UNESCO International Rising Talent Award. Dr. McAfee now serves as a research associate at UBC and studies queen overwintering strategies, pheromone biology, and drone heat tolerance. As an author, she has published magazine articles in The Conversation and Scientific American and writes regularly for the American Bee Journal when she isn't fishing, hunting, hiking, or training horses. And I would just say I recommend her most recent article in the American Bee Journal, Breeding Better Bees at Scale, as we think about bee breeding and genetics. I know that's something that's really interesting for our club right now, is thinking about genetics in our queens. So please join me in welcoming Dr. Alison McAfee. Thank you very much for being here, Ali.
Thank you so much. I hope it's okay that I kick you off the screen
Yeah,
sharing.
yep, I'm going to stop mine and turn it over to you.
Perfect. Okay, let me just get organized here. All right. Thank you so much for that introduction. I really appreciate that. And the shout out to my recent ABJ. That might be my last one for a while, actually. I'm kind of stepping back from that. But I think I left it on a high note. I like that article. I think it was some important information to get out there. So thanks for acknowledging that. So how many of you, when you've opened up your colony, have seen something like this, where things look mostly as they should? It's not a 10 out of 10 brood pattern, but it looks okay. There's no visible disease that's obvious. There's a decent worker population. They have food stores, etc. But there's this queen cell here in the middle, which is clearly a supersedure cell and not a swarm cell. So 8 years ago, when I started researching queen failure and queen quality, I probably, if I saw this, would cut out that queen cell and move on. Maybe just hope that when I came back to it, they would sort themselves out, and eventually give up on trying to supersede that queen because to my eye, it looked like she wasn't doing a terrible job. But now, with some more years of experience and research to have learned some things, I think I would handle this fairly differently. It's pretty clear to me now that when you see supersedure cells like this in the colony, the bees are doing it for a reason, and we should be listening to them. So that's kind of a classic example of what we'd call a queen issue, where there's something, it may not be obvious what it is, but something is causing the queen to be superseded prematurely. And by prematurely, I just mean before you would kind of expect her natural demise. So she's not a super old queen, and those bees were not on the brink of swarming and trying to replace a queen naturally for those reasons. There's something kind of happening or has happened to her to make this to make the workers want to turn her over kind of before she would normally expire. And queen issues like that are quite a common occurrence. This is some survey data published by the Bee Informed Partnership, where they found that queen issues were among the top 2 or 3 most frequently reported causes of colony losses for sideliner beekeepers and commercial beekeepers, for overwintering losses. And it is also very common in the summer, where it ties with varroa mites and associated viruses for being one of the top 2 most commonly reported causes of colony losses there. And as may become apparent in the rest of my presentation, that might not be a coincidence. Why these 2 issues, varroa and viruses and queen issues, are kind of traveling together in terms of the frequency that people are reporting them as a major concern. So that convinced me that this is an important thing to be looking into. It's such a common problem, and we know so little about what its origin is. So in that very paper written by Selena Bruckner, she wrote, "It remains unclear what exactly causes evidently good queens to fail." And by this time, when this was published in 2023, I had already been researching queen failure for some time, but this really underscored to me that it really was still a mystery, and it really is an important thing to try to get to the bottom of, is what is causing good queens to fail. So in our kind of logical framework for what ended up being a very, very lengthy and multifaceted project, we wanted to do 3 main things. And all of these things were done in collaboration with my colleague Abigail Chapman, who is actually, just as of a couple weeks ago, she's the freshly minted provincial apiculturist for British Columbia. So that's very exciting for her. But her and I worked on this together almost every step of the way. So I really need to acknowledge her contributions here. So first, we just wanted to start by gathering queens from beekeepers, and we wanted those queens to kind of fall into 2 groups, to either be failing queens that beekeepers have perceived through their own ways, which was diverse, that the queens were not performing as they would expect, or say they were laying drones, they were being actively superseded, or maybe they were determined to be failing for some other reason. But basically, we just wanted poor queens as determined by the beekeepers and healthy queens to compare them. And then we wanted to do some autopsies. I call them autopsies, but it's not, they're not, we know how they died. They all died by decapitation by scientist. But it's an autopsy in the sense that we wanted to look at causes of failure rather than causes of death. And so once we had that survey data, so kind of seeing what is going on the ground, we could generate some hypotheses around what might actually be causing the queens to fail and do some experiments to address those. Because with any survey, it's very, very difficult to actually draw that causal link between two things that you're observing. You really need controlled experiments to isolate those factors. So do a survey, generate hypotheses, do some experiments to see if they stand up to some real tests, and then take those queens from the lab back out into the field and see if we could kind of reconstruct the queen failure situation in actual colonies. So that's kind of the framework of this journey that I'll take you on. So for these surveys that I'm talking about, there are a whole suite of different things we could measure. These are all, and this is not an exhaustive list. These are all different queen quality metrics. There's queen pheromone profiles. There are physical metrics like her head width, thorax width, wing length, basotarsus length, things like that. There's her ovary mass, ovarial number, spermatheca size, sperm count, sperm viability, mating frequency, which is the number of drones that she's mated with. There are a whole bunch of different things you could measure to measure queen quality. We decided to focus on just a couple things because we had like 180 queens to get through. So we kind of focused our efforts on just looking at their ovary mass and their sperm parameters, because to us, these were kind of the two things most directly linked to their fertility and reproduction. And then we also measured viruses. So we used their heads and measured virus levels there. And we also wanted to look at the nosema abundance. So we submitted their heads and guts to the National Bee Diagnostic Center up here in Canada to measure these pathogens for us. And I will say, if you wanted to know like the real nitty-gritty details about these surveys, we do have published data in these two publications here that are publicly available. But suffice to say, the main findings here were that the failed queens were worse off for almost everything that we measured, except for nosema. There was no difference in the nosema levels between the failed and healthy queens. But the failed queens did have lower sperm viability, fewer sperm stored in their spermatheca. They had smaller ovaries, and they had higher levels of some viruses compared to healthy queens. Not every virus, but when you took the whole panel of viruses that we measured and added their copy numbers together to get like a total viral load or like a more complete viral burden parameter, the failed queens had overall higher viral loads than the healthy queens there. But then the thing that really interested us was that when we looked at relationships between these pathogens and these fertility metrics that we measured, we didn't see a relationship with the sperm parameters, but we did see a significant relationship with the ovary mass. So the queens that had higher viral burdens had smaller ovaries. So that really jumped out to us as being an interesting relationship to follow up on, because the kind of natural hypothesis that falls out of that is, well, maybe viruses are causing the queens to have smaller ovaries and are the culprit for queen failure. But like I mentioned before, you really need experiments to be able to show that, because maybe the queens with smaller ovaries in our survey just had smaller ovaries because they're dud queens and they're not being fed as well. And then when they don't get as good nutrition, that makes them more susceptible to a virus infection, something like that. There could always be some other meandering way that you could arrive at a correlation without having a direct cause. So we did kind of a quick and dirty experiment to address that. We had the hypothesis, of course, that virus infections were causing the ovary mass to decline. And we tested that by buying queens. This is what, a couple dozen queens from a commercial source. And we split them into two groups. And half of the group got injected with Israeli acute paralysis virus, which just happened to be the only virus that we had in the freezer right then. And the other group was injected with saline. So that is controlling for the effect of injury that could conceivably have an impact on their ovaries and well-being as well. And if you've never seen a queen's ovaries, I just wanted to show you some pictures of the dissections at the end of this. So after 2 days, we took all the queens in both those groups and dissected out their ovaries to weigh the mass of them and measured their virus infection to see if we again got that same negative relationship that we saw in the survey. So this is a queen with her dorsal plates removed, and that's her ovary exposed there. And I just want you to appreciate how ginormous of an organ that is, and to kind of put it into perspective, the resources that are required to maintain those ovaries at that size. It's like a third of her body weight. So it's really a massive amount of resources that she needs to put in to maintaining those, keeping them active and being able to push out eggs. So that's just a picture of one with the ovaries and one with them dissected. And then when we measured the virus levels here, so the further to the right on this axis we go, the higher the IAPV virus load we see. We found that there is the same negative relationship between virus mass virus infection and ovary mass. So here we have ovary mass on the y-axis, and the queens with the largest ovaries are over here with the lower virus levels, and the queens with highest viral levels have the smallest ovaries. And that was a significant relationship. So that kind of told us that we were on the right track, that maybe we were onto something. Maybe this virus relationship is more than just a coincidence, but it also wasn't the greatest experiment because IAPV is a pretty nasty virus. It makes the queens really sick. So at the end of this 2-day period, when we sacrificed the queens, they were shaken, like they were showing symptoms. I worried that maybe what we were seeing was just an effect of them not being able to eat as much as the healthy queens. So we wanted to do a better experiment. So we upgraded our cages. For one, we started using these queen monitoring cages, which were generously shared with us by Julia Fine, who's down at UC Davis, I guess, kind of in neck of the woods, at least in the same state. These are really cool cages because you can keep the queen and around 100 workers in them. And you may see in the back of the cages, there are these hexagons. Those are egg-laying plates that you can actually remove and replace with fresh ones. So it lets the queen kind of do her queen thing. She can be in there, lay eggs, and you can count the eggs in a set period of time to actually get a clear metric on her egg-laying output. And we also upgraded the virus situation. So instead of using IAPV, we used a mix of deformed wing virus B and black queen cell virus because those were two of the most abundant viruses that we saw in that initial survey that we did. And they're also two viruses that don't cause severe pathology in adults when you infect them. So it's avoiding this problem of like the IAPV queens being so sick they couldn't eat It's kind of getting around that by using a different, less intense virus. And here we had a hypothesis that was a little bit more complicated. So we thought it was possible that virus infection could cause a reduction in ovary mass, not just through direct pathology of the virus. So the virus is like killing the ovary cells, for example, but just through the very fact that they stimulate the queen's immune system. This is something that has been shown in other insects as well, where just giving an animal an immune stimulation can take resources away from their ability to invest in reproduction. And just given the huge size of the ovaries that I just showed in the last slide, it's pretty clear that they really need to invest a lot in their ovaries. So it seemed at least feasible to us that shunting any resources away from that process by stimulating their immune system could be having an impact on their ovaries alone. So to be able to test that, we needed a little bit of a different experimental design. We had three groups this time. So we had queens that were all introduced into these cages and laying eggs, hunky-dory, everything's good. And then we take them out and do one of three things to them. We either inject them with a mixture of black queen cell virus and DWVB, like I mentioned, and so that's our infection group, or we inject them with the same mixture of viruses, but where the virus particles have been irradiated with UV light, so they're no longer viable viruses. They can't cause an infection. They're just going to be able to stimulate the immune system because of these foreign particles that you're injecting in, but they can't actually reproduce. Or we have the control, like before, of just a saline injection to control for stabbing them. And then we put the queens back in their cages and monitored them for 1 week to look at whether they restarted egg laying, and if they did, how many eggs did they lay. At the end of that period, then we sacrificed them and measured the levels of viruses in them, weighed their ovaries again, and conducted proteomics on their hemolymph. And that's just a method of measuring that we used for measuring their immune stimulation in their blood to just see if their immune systems really were activated or not. And what we found with the egg laying was really interesting. So here, the kind of olive color is indicating that the queen did restart egg laying after we put them back in their cages, and black is indicating that they did not. So for the control group here, this means that 8 out of 9 queens did lay eggs again after being injected. 1 queen did not. So most of them started laying again, which is what you would expect. The queens getting the UV inactivated virus mostly started laying eggs again as well. A couple of them didn't. The queens that got the infectious virus, they mostly did not start laying eggs again at all. So this is like in that 7-day period after being injected, not a single egg was laid. And that was a significant result. So that was really interesting to us. It really did look like the virus was having an impact on her ability to restart egg laying, at least in this time frame. Probably if we went longer, they would eventually be able to lay some eggs, but this experiment only lasted for a week. Then when we looked at the levels of viruses in the queens, I guess I didn't want to show too many graphs because there are already a lot of graphs, but just maybe trust me when I say that we did our due diligence to really check that these queens that got the infection really did have higher levels of viruses compared to these two, the control and the inactivated group. So we did show that. And then when we measured their ovaries, we found that the control queens, as you would expect, had the largest ovaries, And the queens getting the virus, the active virus infection, had significantly smaller ovaries. They're like maybe two-thirds of the size. And the queens, interestingly, that got just the virus particles that couldn't actually cause an infection, they it wasn't as big of an effect, but it really looked like there was an impact of that. It was not quite statistically significant, but there's a pretty clear trend that the queens in that group had tended to have smaller ovaries than the control queens. So what this told us was that just the act of stimulating the queen's immune system alone was doing something. It was partially, but not entirely, causing this reduction in ovary size. So then when we looked at the immune protein data from their blood samples, we found that yes, both of these groups did have an elevated immune response. So what you're looking at here is a heat map where each column is a different queen and each row is a different immune protein. We actually measured thousands of other proteins as well, but I'm just showing you these immune proteins because they're the ones that are most relevant to this topic. So the blue color represents a low infection, so there's not very much of, or sorry, low abundance, so there's not very much of this protein that we found in the blood, and pink is higher abundance. So the queens that just got the inactivated particles had a higher immune response than the controls, and it was very similar to what we saw in the infection group. So yes, both of these injections stimulated their immune system. And we think it is a reasonable conclusion that they that the that the infection did oh, I've lost my words. That they the that this immune stimulation is partially but not wholly responsible for the effect on the ovaries that we saw. So what we've learned so far is that in that survey, we found that the failing queens did consistently have smaller ovaries than healthy queens, and that virus infections did reduce the queen's ovary mass. And now across a number of different viruses, we had IAPV in the beginning, and then the DWVB and black queen cell virus mixture that I showed second. And this also inhibited their ability to start laying eggs again. And that the effect is partially, but not wholly, just due to that effect on the queen's immune response, maybe shunting resources away from her investment in reproduction. So I just wanted to pause for a second to see if anybody had any questions thus far before I dive into the second part of this presentation. If there aren't any questions, then I'll move on.
I
But
have a question.
yes,
Would you say that the viral load that you added is sig is at a quantity that a beekeeper that can visualize virus in their colony, observe, greasiness or behavior, would you say that your viral spike was at a level that beekeepers could see a viral activity in their colony, or would you say it's below that level?
that's an interesting question. I would say it's probably below that level. So we used a dose that was trying to match about what we were observing in the failing queens in our survey data. And when we received those queens, they did not look sick to us. We couldn't see any symptoms in them before we sampled them. They did come to our lab live, so we got a pretty good look at how they were. Um, yeah, we couldn't see any symptoms to our eye. So I don't think that it's something that you would be able to see on the ground either.
Thank you very much.
Mhm. Good question. Okay, if there aren't any more, then I'll move on. There will be more chances for questions at the end, of course. So that kind of covers the first two parts of our big project. But really, I think the big questions are when you take queens, infect them, and then put them back in colonies in the field, do you see the same thing? Or is the kind of colony superorganism able to mitigate those effects that you're seeing in this stressful lab environment? So what we did to address this was I made up 32 five-frame nucleus colonies, and I thought it was very funny driving around the city in my RAV4 with all these nukes in the back of my car, but that is what I had to do the job with, and we got it done. I made up all these colonies and then split them into two groups this time. So half of them, once the queens were introduced to their colonies and they were accepted and laying eggs. Half of them we took out again and injected them with the same virus mixture, the DWVB and black queen cell virus that we used in the cage trial. And half of them were again injected with saline. And then they were put back in their colonies and everybody was mixed up. I didn't do the injections, so I didn't know who was in which colony. And I took a bunch of measurements. So every other week, I would record the number of supersedure cells. I'd weigh the colonies. I'd check their food. They had pollen patty and fondant on all the time just to make sure that like foraging differences couldn't cause someone to have really poor food stores compared to another. I took photos of each brood frame to measure brood area and adult bee area and counted the number of disease cells that I found if there was a case of disease. And 2 months later, I sampled the queens and sampled the workers to measure the virus levels in both. And then also dissected the queens and weighed the ovaries and her virus levels to compare the same parameters that we compared in the lab. So the first problem came when we looked at the viruses in the queens and the viruses in the workers. So on this graph, I have the queen virus levels here, where higher numbers obviously are higher virus levels, and then the worker virus levels here on the x-axis. And what we thought we would see was that the queen viral load would split into two clear groups where the controls would have less infection than the ones that we infected with the viruses. But that's not what we saw. We actually have some control queens way up here with super high virus levels. And it's pretty clear, I think, from this graph that there's a relationship with the worker virus levels. So what happened was that our injection, which remember happened 2 months ago before these queens were taken, was swamped out by the workers transmitting viruses to the queens when we put them in the colonies. So that was a bummer. But it did also show us that, wow, colony virus infection dynamics are really important. And yes, it appears the queens can get sick from her workers. But it kind of screwed up the experiment because now we don't have two nice groups since so many of our control queens have high virus infections. And likewise, many of our infected queens actually seem to have recovered down here when they were put in really clean colonies. So this did turn into less of an experiment and more of like a glorified observational trial. But it was still really useful because all these nukes were made at the same time. They were kept in the same yard. They were given the same feed. They even had queens that were produced from the same graft from the same producer at the same time, etc. So like it was a really controlled environment with fewer variables differing between the colonies than in, say, that first survey that we did where we just took any queens that beekeepers were willing to give us. I guess the moral of that story is that queen virus infections reflected the worker infections, not what we actually injected them with. Um, but despite all that, so when we just ignore which group we had intended the queens to be in the beginning and just look at the level of virus infection that they actually had, we again see the same relationship, the same drop in ovary mass when the queens have higher infections. So uh, that was really interesting to us because it's showing us that it's something that can still happen under these controlled conditions when the queens are in real colonies. And then I mentioned I measured all these other things too, but I'm really just going to focus on the supersedure cells because that's what I think is the most important. And here on the y-axis, this scale is really just the probability of observing a supersedure cell. So we go from having no probability of seeing a supersedure cell in the colony all the way up to 1, which is like complete 100% probability. And when we plot whether we saw the supersedure cell in the colony, whether we saw it in the colony or didn't see it in the colony in relation to the level of viruses that we measured at the end, there's a really clear upward trend where there's a higher probability of seeing a supersedure cell in the colony when the queens are infected. So supersedure cells are positively linked to their viral load. Really, really interesting to me. So that really, this whole experience was just like following a trail of breadcrumbs where one piece of data just leads to another question. And here, when I saw this result, that immediately made me wonder if the virus infections were changing something about the queen's pheromone profile, because the queen's pheromones are so important for signaling in the colony, her reproductive status. It it causes like a whole suite of changes in the worker's physiology and behavior. That's kind of like the central thing that would make the most sense um, as something that could be impacted by the viruses to change the worker's behavior and shift them towards wanting to rear a supersedure cell. So the queen pheromone, I don't know how many of you are really familiar with it, but it's not actually just one compound. It's really best thought of as a whole bouquet of at least 9 different compounds that make up the queen retinue pheromone. And some of those are made in her mandibular glands, and some of those are made elsewhere in her body, and we actually still don't know where they are all made. But luckily for me, we had just come up with a new method of measuring these compounds, the queen pheromone components, in our lab. And so this was the perfect research question to apply it to. So I took all these queens we had now, the ones from the cage trial and the ones from this field trial that I just told you about, and measured their pheromone profiles. And when I looked at their abundance compared to the level of virus that the queens had, there was one that popped up as being related to both methyl oleate. So methyl oleate is not a component of the queen mandibular pheromone, that the classic one that is made in her mandibular glands. It's one of the ones that's made elsewhere in the body, and we have no idea where it actually comes from. But it was clearly linked to both the virus level and the ovary mass of the queens. So queens that had heavy virus infections and little ovaries had lower methyl oleate. Queens that had very little virus infection and big healthy ovaries had a lot of methyl oleate. So this really seemed like a good candidate of a signal that the workers could theoretically sense that could tell them that the queen had a virus infection and that she maybe should be replaced and stimulate supersedure. So there was one problem with that, though. I didn't know if it was the virus that was causing the reduction in methyl oleate or if it was the shrunken ovaries, because those two come hand in hand, right? The virus causes the small ovaries. So which one is it that's actually changing the abundance of methyl oleate? So I did another experiment where instead of infecting the queens, I shrank their ovaries by caging them. So if you cage the queens, then she has nowhere to lay, and her ovaries will just kind of atrophy as a result of that without any change in infection. And when I measured the pheromones from them, again, methyl oleate was the only one that was reduced. So it was clearly an effect of the change in her ovaries and not a result of the virus infection. So that was really interesting to me because it's like this could be kind of a broader mechanism of supersedure whereby anything that causes the queen's ovaries to atrophy, which would imply that she's no longer able to lay sufficient eggs, is causing this change in methyl oleate and a signal that the workers can sense. So those are big claims, but again, I need to do an experiment to show that methyl oleate actually is important for inhibiting supersedure for any of that to make sense. So that is what I did. But first, to back up for a second. So where we're at right now is that we kind of have this logical model where it looks like the virus infection is causing the queens to have shrunken ovaries and reducing their laying. Then that impaired methyl oleate production is something the workers can sense to possibly cause supersedure. So to actually test this, I did another experiment where I had 30 nucleus colonies again, and they were all queenless. They all had, however, a frame of eggs and ample food, a good worker population. So they had everything they needed in theory to be able to rear queens. And then I added back different pheromones to each of these groups. I had one group that, of course, was the one that got no pheromones added back, and that was the control. So in this group, you'd expect them to just rear queens because they're in that normal emergency queen rearing situation. Um, then I had a group where I gave them back queen mandibular pheromone, which does not include methyl oleate. And we know that it has some effect on inhibiting queen cell rearing. But what we really wanted to know was if you add methyl oleate to that, does that further inhibit the queen cell rearing of the workers? So what I expected to see when I came back to these colonies 2 days later was to see lots of queen cells reared in the no pheromone control, the least queen cells reared in the ones that also received methyl oleate, and the ones receiving QMP would be somewhere in the middle. I wasn't sure where. And that is surprisingly exactly what we ended up finding. So whether we looked at the number of active cells, so that was just a queen cup that had royal jelly in it, I called an active cell, but not necessarily had a larva. Sometimes they just wet the cells like they're getting ready to rear in them, but haven't fully committed yet. Whether we looked at that as a metric or the number of charged cells, which was a queen cell that had the royal jelly and a larva that was like fully committed to trying to rear a queen here, or just the overall frequency of colonies that had any active queen cells at all. We saw the same pattern where the control group had more queen cells. The group receiving QMP plus methyl oleate had the least, and the ones just receiving QMP were in the middle somewhere. So this was really, really exciting to me because, well, first of all, because the whole thing was done blind. So I had no idea which ones actually got which treatment. until after I had analyzed the data and I got my colleague to actually tell me which was which. So this was kind of an exciting reveal because what it shows is that methylfolate is contributing to some degree to this inhibition of queen cell rearing, being consistently the group that had the lowest number of reared cells. So at the end of all this, I guess we always, as beekeepers, want to know, what does this mean for you? What does this mean for your colony management? I've kind of figured out this really interesting mechanism, I think it's interesting, of how queen virus infection can change her physiology and how workers can sense that. But on the ground, like, what should you do differently? So one piece of advice, if you see a colony like this that is playing with supersedure cell rearing, if you cut the cell and replace the queen, it'll buy you some time, but it won't actually solve the underlying problem. So if the bees are rearing a new queen because they've detected that there's a virus infection, then if you put a new queen in there, she, in a couple months, like I saw in our experiment, she'll probably just become infected again because the workers are all sick too, even if you may not be able to see it. And then you'll just be in the same position 2 months later when you might not be in as good of a place to be able to fix it. Say this happens like late in the summer, and then a couple months later, you're already into the fall and you might miss it. Or if they try to requeen naturally with another supersedure event, they might not be successful. So it'll buy you time, but it won't solve the problem. I think if I were to see this now, what I would do is treat this colony for Varroa, because we all know that Varroa is such a good vector and amplifies some of the viruses that infect the bees, especially DWVB. So I would treat them for Varroa to just try to break that cycle and then requeen. So try to give the bees a chance to overcome that virus infection and then introduce a new queen, and that might give you a better chance. We're also looking into new ways now, this is kind of our current research, of producing nukes that can limit virus transmission. So the idea that we're working on is, can we improve the colony health by producing nukes, not by the normal method of splitting with brood frames and food frames, but by just doing like the shook swarm method, just shaking bees onto foundation and feeding the heck out of them to let them build their comb up, treating them for mites with oxalic before they hit that point where they've capped any brood. And then that gives them, the idea is that should give them a really good foothold starting out on fresh comb that we know viruses can be harbored in comb and be transmitted that way. So they have fresh comb, you knock the mites down, give them a chance to overcome those virus infections, and do all this in July. And then that, in theory, should kind of set them up for success over winter. So that is an ongoing project that I don't have data on yet, but we're pretty hopeful that that can make a difference. But if I can leave you with just one message after all of this, it is, when you're doing your mite treatments, think about doing it for the queen instead of just doing it for the worker bees. You're really helping to protect the queen as well. I think that, I don't know about down there, but up here, we're having a number of discussions around whether we should actually reduce the treatment threshold from 3% down to 1% throughout the year. So up here, again, I don't know what your norms are in California, but up here, it's pretty typical to use a 1% treatment threshold in the spring and a 3% treatment threshold in the fall. But based on what I've seen, I think that we really need to uh, to limit Varroa earlier than that, because what ends up happening is in the fall, you can have these colonies that are hovering just below 3%, and you're, oh, okay, I don't need to treat. So you don't treat, and then all the drones get kicked out, and all of a sudden you have like skyrocketing mites on the workers because they used to be on the drones and now they're on all the workers. And then it's like a panic to treat. You're approaching fall. The mites are at such high levels that they're really vectoring and amplifying the viruses. Probably you've sent them on this upward virus trajectory right at the time when it's most critical for them to be healthiest. Because if the queen does get sick in the fall or winter, there's, at least up here, maybe your winters are mild enough that you can still manage things in the winter, but up here we can't. And if they get sick in the winter, then there's nothing that we can do about it. They will go on undetected. Maybe the colony will try to supersede and obviously fail because there aren't good mating conditions then. So when you're treating for mites, think of the queen, don't just think of the workers. And maybe we can have fewer colony losses because of these poor queen issues. Thank you. I'll take other questions now.
Thank you very much. We'll open up to questions. And if folks have questions also that they want to write in the chat, we can write them in the chat and I can read those out as well. So thank you very much.
Yeah, no
Okay.
problem.
I got a question. So how often should we replace the comb?
How often should you replace the comb?
Yeah, if the viruses are staying in the comb and it can be passed on, should we start with fresh foundation and let them build out the comb every year?
Yeah, I definitely think we should be doing that more often than we are. I mean, the sort of standard recommendation is to cycle your comb out every 5 years. So that means, like replacing 20% of your comb every year. But I don't know. I don't know if that should change based on this or not. But I do know that I think we're better off doing it more often than we are. I just don't have the data to really like back up saying, oh, we need to replace it all every 3 years or something like that. Um, First, we'll see if we do see a positive impact of this experiment where we are shaking the bees onto foundation and wintering them on fresh comb. If we see that that is helpful, then I'd be a little bit more comfortable giving like a hard recommendation for that. But until then, it's still just a hypothesis. But we do know that the viruses can persist in the comb through the winter. So, it's always a trade-off because comb is expensive to produce, right? And it's a valuable resource. If you give them drawn comb, then they can get to work on it so much faster than if you give them foundation. So there's a situational component too. But at least if you know that there might be a trade-off to using that older comb, then if your situation allows to use foundation or fresher comb, then I would definitely encourage that.
All right. Thank you.
I have a question about since the ovary size really matters, and with smaller ovaries having less methyl oleate and not the bees having less pheromone. So when we see a skinny queen, do we assume the ovaries are a lot smaller? Or sometimes you have a super fat breeder queen, she's all swollen. And other times you have a skinny queen and she's doing excellent. Do you have any comments about the size of ovaries in a smaller queen versus the larger
Yeah.
ones?
Yeah, that's tricky because they really do have they come in all shapes and sizes, don't they? I've seen what you're talking about. I've seen queens that are like I call them long queens rather than skinny queens, where they just have abdomens that are like, whoa. Um, yeah, it's really hard to say just visually whether whether she has small ovaries or not. I think you would have, I think if you have the ability to observe changes in the queens over time, then you might be able to tell. But I don't have confidence in my ability to just look at a queen and kind of guess at her ovary size, just because their abdomen shape does have so much diversity to it.
I had a quick question. You mentioned how caging a queen can result in atrophy of the ovaries. Do you know how long a queen can be caged before we start seeing that atrophying of the ovaries in a way that might affect the level of methyl oleate and her sort of acceptance by the bees or the drive to supersedure?
Yeah, that's an interesting question. There are lots of questions in there, actually. So, one is how long can you cage them before you see this effect on the ovaries? And uh, about maybe a few days, and you'll see a change in her ovary mass just from being caged for a few days. Now, that depends if she's caged with a few workers in the cage and taken out of a colony, or if she's caged and put in a bank. Then you see less of an effect if she's put in a bank because she is still getting a lot of food from so many workers still interacting with her. But there is still an effect. It's just less of an effect. So in either situation, in within a few days, you'll see shrinkage of the ovaries, but that doesn't, first of all, that's not a permanent change. So they will bounce back once she's introduced to a colony and gets better fed, gets laying again. Then they will grow again and become as normal. Um, and also, we did an experiment with Shelley Hoover. Shelley, actually, she was the one leading it. And she tested introduction success of banked queens versus queens that had just stayed in their mating nukes for the same period of time. And there was no change in introduction success. So this methyl oleate thing seems to really be specific to supersedure. Like once a colony is there and laying, and then there's a change in methyl oleate, then that can trigger the supersedure. But when you're just introducing a queen for the first time, um, there wasn't any impact. And those banked queens that went into the colonies, they had smaller ovaries, but they were accepted just as well, if not better, than the ones that weren't banked.
Thank you very much. There's a question in the chat. Based on your findings, are all viruses associated with reduced ovary size in queens?
All the ones we've looked at, but we have not looked at all the viruses. Yeah. So we've looked at IEPV and DWVB and Black Queen cell virus, as you saw, and all of those had an impact, but there are many, many other viruses that we just haven't tested.
I have a comment. Our club in next month is buying a lot of the Golden West queens for members. They can buy them, and so it sounds like you would recommend that members treat their colonies well for Varroa before any requeening of these supposedly hygienic bees. Is that correct?
Yeah, I think if you have reason to suspect that you have a virus issue, then I would recommend really treating them before doing a queen introduction. If you say you've been testing your Varroa and all along you've gotten like zeros and ones, and you're not seeing symptoms of viruses either, not that we should rely on that. That's just like if you do see symptoms, then you know you have a problem. If you don't see symptoms, you might still have a problem. But um, yeah, I'd say if you're getting zeros and ones in your mites and you're not having other reason to believe you have a virus problem, then you probably don't need to worry about it. Uh, but for a colony like this one in the picture, if I'm seeing supersedure cells, then that's like giving me reason to think and maybe do something differently before just replacing the queen.
But in any case, make sure your mite levels are under control.
Yeah, it's also a really good opportunity. If you know that you're going to be replacing the queen, like a lot of people say that they see like queen loss when they treat with formic acid, something like that. So um, if you know you're getting a new queen in not too long, then you can just nuke them with formic acid and you don't have to worry too much because you've got one on the way, right?
For sure. Thank you.
Yeah.
Another question on packaged bees. Is it a good idea to treat with maybe oxalic acid vapor?
Yeah, so whenever I get packaged bees, I treat with oxalic as well. Personally I do a dribble, but I think that both would be fine. Certainly, it's a great opportunity when there's no brood. Just yeah, take that opportunity to treat them with oxalic. I wouldn't even test them for mites. I wouldn't take those 300 or so bees away to check first. I would personally, I just whenever I install a package, I just do a dribble and um yeah, that's like my standard procedure. Might as well knock them back while you have the chance.
Sounds good.
Because even the best package producer will have some that have mites that get through, right? It's always gonna happen once in a while.
There's a question in the chat, kind of following up on the comments you made about treatment thresholds and treating at a certain threshold. And the question is, what happens if I treat for mites and I actually don't have any mites? So that, what is the risk or the danger involved in applying a treatment when perhaps there's not an actual mite issue to be solved?
Yeah, yeah. That's definitely something that's important to think about because there is a trade-off with pretty well every treatment out there, right? Formic acid one is the easiest one to kind of use as an example because when you do the 14-day treatment with say two Formic Pro pads, you're not supposed to go into your colony a few days after, but if you ever have, then you would see that like all the open brood is dead. Um, so for sure, there's a trade-off. I'd say that yeah, actually that's a good point. So don't take what I said to mean like just treat all the time. Certainly, I think we still need to use thresholds. So I think based on current knowledge, and this could change, I think that 1% is a reasonable threshold. Um, But yeah, don't take that to mean that you should just be treating like every opportunity that you get. Often, actually, I don't know off the top of my head for every single product, but certainly for some of the products, there are on the label uh, limits to the frequency that you should use them. So follow those as well.
Great. Thank you very much.
Mhm.
Any other questions?
Have you heard about that new product on the market in Aurora?
Yeah, I'm really excited about it. I think it's super cool. Um, I saw some data presented by one of the tech transfer teams up here in Manitoba, who is working with Greenlight Biosciences to do some field testing for it, and it looks really promising. I don't remember the numbers off the top of my head, but a single treatment early in the summer was enough to keep the levels uh, below the current treatment threshold of 3% into, I believe it was the beginning of September. So that was pretty awesome, I thought. Whereas the ones receiving uh, Apivar were way above that because resistance is such a problem now. And obviously the colonies that didn't get treatments were even more above that. So there was a really clear effect, and it seemed to last for a really long time, which is what I was excited about.
Yeah, because I tried it in one of my hives, and it looks promising.
Awesome. Yeah, that's really cool that it's come to the market so quickly. I think we're still waiting on it up here. I'm not too sure. We're usually slower than you guys. I think with the exception of formic acid, maybe we're slower at getting things approved. Um, but yeah, I think it's super promising.
Okay,
So
thank you.
can I ask you a question?
Yes, go ahead, Dennis.
Well, we're getting these new bees that are supposed to be potentially resistant to the varroa mite. How does that work exactly?
That's a big question. There are many, many different mechanisms of varroa resistance. I spent my PhD studying one of them, which is hygienic behavior and varroa sensitive hygiene. Um, but there are lots of other behavioral mechanisms too. They also can uh, be better at grooming mites off one another. So that's grooming behavior. Hygienic behavior and VSH, those are both where the bees, the workers, can detect that a brood cell has an infestation, and then selectively remove or disrupt the mite reproduction within that cell. Then there's uncapping behavior, which is where they just like open and close the cell. And for some reason, that is sufficient to stop the mite's reproductive cycle as well. So those are three. And then I know I'm forgetting a bunch of them, but suffice to say, there are lots of different behaviors that the bees can do that will limit varroa. There's sort of like the super duper groomers from Purdue University, those mite biting bees that they don't just like knock the mites off their sisters, they also like really damage them. Um, so then they can't crawl back up. That's kind of a fun one. But yeah, I'm sure you'll hear a lot more about that kind of thing in Marla's talk, I assume, because she's kind of the godmother of hygienic behavior. I don't know how much she would touch on the other behaviors, but all these are things that can be bred for. It's just really difficult. Any breeding consistently for any trait with honeybees is difficult, as I'm sure you all know, because it's just so hard to control the matings, and make sure that your queens are mating with the right drones, but not mating with too many of exactly the right drones. So you still have enough genetic diversity. That's been like a formula that has been extremely difficult to pin down. Um, but some people have had success. So,
So,
um,
so as a follow-up, how do wild hives handle this problem?
multiple ways. So one of them is just by swarming more. So I think that is a really big one because when a colony swarms, they obviously need to build new comb when they arrive at their new home, right? So they have a period where they have totally fresh comb, where they have no brood, so they get to like they have more grooming opportunities. Um, because there's no brood, the mites can't be amplifying at that particular time. So they have a bit of a lid put on them um, from that as well. So all these things really slow down varroa and slow down the the viruses that they vector as well. Even this is something I've just learned recently, but even just not having brood changes the bee's immune system. So changes the worker bee's immune system. So they get stronger immunity and can clear out those viruses more effectively than than just having the presence of brood. Something about those pheromones and how it impacts their physiology, I don't really know. But that is a really compelling natural way of limiting the varroa growth. So that's another reason why we're really keen on this new idea, new method of nuke production. Is it's kind of like mimicking swarming. And yeah, I'd say that's probably the main reason. They might also have more selection, natural selection towards things like hygienic behavior. But if I were to guess, my opinion would be that probably the swarming frequency is having the bigger impact.
That goes with getting new comb.
Exactly.
Thank you very
So
much. I second the plug for Marla Spivak's talk. I think she's going to be talking about all those different hygienic behaviors in April for the Zoom event.
Yeah,
Yeah.
that's awesome that you got her to talk. I guess she's retired now, so she has more time or something.
Yeah.
I don't know. Yeah, she was kind of my little bit of my idol when I was a student starting out, and she ended up being the external reviewer on my PhD thesis. So
Nice. Yeah.
yeah,
Yeah.
some
Well,
fun facts there.
she's a legend. So, yep.
Yeah, yeah, exactly.
Yeah. Any other last questions? Well, thank you very much, Dr. McAfee, for presenting. It was a great presentation. We really appreciate you taking your time to come and join us this evening.
Of course. Yeah, it was really great to share it with you. This is the first time that I've shared it kind of all together because this is literally like 8 years worth of work. And it was only last summer that I did those pheromone trials. So it's the first time that I've kind of tried to tie it all together. So I hope it left some points stuck in your mind and that it wasn't all just a blur.
Absolutely.
It did.
Thank you very much. Thank you.
Great presentation. I learned a lot tonight. Thank you so much.
Awesome.
Thank
That
you.
was really
Thank
great to
you.
hear. You're welcome. That was my pleasure, guys. If you
And
have any
hope,
other
hope we
questions
see you
for
in
me,
person one day.
yeah, yeah. If not, on my very first slide, I had my email, and I think that this is recorded, so you can go back and look at that. If any of you had follow-up questions for me later, just drop me a line.
Yep, the recording will be available, and yes, absolutely, Dr. McAfee. If you end up coming down and making a field trip down to Orland or something like that to visit the queen breeders and things, let us know, and
I
we'd be
will. I will
happy to
for
have you.
sure.
Yeah.
Okay.
Okay.
Thank you.
Okay.
Thanks, everyone.